page. And my name is Brian Carpenter. I'm Senior Director, Unstructured Technology Strategy for Pure Storage. And I have with me two very important people to talk about not only what we're doing from a technology perspective, but how the teams at Foldinghome really are working on that COVID-19 problem. So Stacie and Greg, why don't you go ahead and introduce yourselves?
Yeah, Stacie Brown. I'm a field solutions architect here at Pure Storage. I'll pass it over to you, Greg.
I'm Greg Bowman. I'm a director of Foldinghome and an associate professor at Washington University School of Medicine in St. Louis.
And one of the reasons why we're here today, Greg, is through a series of very fortunate events. Some industry partners of yours and ours sort of introduced us in the very early portions of the kind of COVID-19 pandemic. It was over a year ago. So it's you've gotten younger, I've gotten older. It's been a great year. Through that introduction, not only do we get to learn about what you're doing, and Foldinghome has been around long before the COVID-19 pandemic, and and all the things we're trying to do here, but also the things that you did through the last year are obviously very impressive. So that's why we want to talk today. It is a great opportunity, Greg. Did you have anything to say real quick?
No, this is amazing. We've been having a lot of fun working with you, so appreciate it.
Yeah, thank you. And Stacie, why don't you tell everybody, obviously there's probably a lot of people who have Foldinghome installed, but maybe they don't have Pure Storage installed. So why don't we talk about what we do in relationship to what Foldinghome does?
Yeah, in a nutshell, Pure Storage is storing all of the data that Foldinghome is collecting, and we're helping Foldinghome access that data to turn it into valuable information. So part of the computation is, of course, leveraging every person's home computer with the Foldinghome binaries, and then that information gets taken back and stored on a FlashBlade, which is our product that offers object and NFS storage. And then that enables Foldinghome to access that data and do really creative, useful things to help find out what's going on with COVID and how we can help combat COVID.
Yeah. And Greg's job is amazing. Our jobs are pretty cool. I mean, getting to wake up in the morning, talking about how we help people with AI and, computational science and all these other kind of emerging technologies. I was just on the phone talking about genomic sequencing, which Greg knows better than both of us combined. But at the end of the day, being able to talk to people about things like ransomware and all those other really interesting things, it's a really fun day to come to work. But it's not as fun. Let's be realistic. It's not as cool as what Greg's about to talk to. So why don't we ask Greg some questions instead of talking about ourselves for a second, Stacie?
Yeah, completely agree. Greg, you've shared before that Foldinghome is not a new company. Brian touched on it earlier. You guys have been around for 20 plus years as a company, and we've heard congratulations are definitely in order for you, as you are going to be in the Guinness Book of World Records for the fastest supercomputer. Can you tell us a little bit more about Foldinghome's mission?
Yeah, it's been quite an exciting ride over the past 20 years, and things have evolved a lot. But currently, we see our mission is really empowering anyone with a computer and an internet connection to become a citizen scientist and help us combat global health threats. And these health threats are wide-ranging. We focused a lot on, what I'll call traditional biomedical health threats, so Alzheimer's disease, Ebola virus, cancer, quite a lot of antibiotic resistance, quite a lot of alarming threats to all of our health. But we've also started branching out into other things that are of a similar magnitude, like increasing the efficiency and productivity of crops, for example, and dealing with pesticide resistance, and starting to think about energy and climate change and how we could play a role there. So it's a really fun area where we get to be at the nexus of a lot of different things, from AI to physics and chemistry and biology and computing and drug discovery and genetics and all of these different areas.
And of course, Greg, you mentioned all these things that you've already been doing, and it really focuses, from my understanding, around simulating protein dynamics and those types of things. But obviously, this past 12 to 18 months, whatever it is, have been really different. So let's kind of dig into what you've really been doing and how you're helping fight the pandemic?
Yeah, so our very general interest, as you said, is in protein dynamics. And for those that hear proteins and are thinking about what meat will be in your entree this evening or your protein shake, really these are the molecular machines that are responsible for most of the active processes we associate with life, right? So it's proteins that allow you to contract your muscles. It's proteins that are detecting light in your eye. It's proteins that are sensing vibrations and in your ear so that you can hear sound. It's proteins that are responsible for breaking down food you eat and building other bits of your body. So they're quite busy and varied in their functions. And when they malfunction, we end up with things like Alzheimer's disease or cancer, and proteins are used throughout biology. So when we think about infectious diseases like bacteria or viruses, a lot of what they're doing is based on these molecular machines, right? And one of the challenges is that, like the machines that we're used to thinking about on macroscopic scales, like our cars and trucks and whatnot, these things have lots of moving parts. But because they're on such tiny length scales, nanometers and lower, we can't zoom in on them with any existing microscope or any microscope that's been conceived really and watch them in motion. We just get these single snapshots of what they usually look like, right? And so if you think about your car, for example, your car is quite interesting. You can do J-turns and donuts and get to work. But lots of times it's not doing anything interesting. It's just sitting in the parking lot. So if you had a snapshot of what your car normally looks like, it's just this big piece of metal parked in a parking lot. And if you didn't know what it was, it doesn't seem all that interesting, right? And so what we do with Foldinghome is we take these individual snapshots that contain an enormous wealth of information, but are far from complete. And we use computers to simulate all of their moving parts and really get into the details of how they work and what's going wrong when they fail to work. And this is of tremendous value if we want to, redesign a protein to do something else or control it, say with a drug, for example, because, just as it's hard for me to think about understanding how my car works and how to fix it or how to make it go faster without seeing all of the moving parts, we're in the same situation with proteins. And so what happened at the beginning of 2020 when it started becoming clear that COVID-19 was going to become a pandemic is that we put all of our varied efforts in Alzheimer's and cancer and antibiotic resistance. And we said, well, these are tremendously important, but there's something new and more important right now. And so we put all of that on side and put all of our intellectual and computational resources into understanding how the protein components that allow the SARS-CoV-2 virus to infect our cells and replicate and spread while evading our immune systems, how all of this works so that we could help inform the development of vaccines and therapeutics.
Yeah, so that sounds to me, Greg, anytime we think about self-driving cars and the amount of data that you're taking similarly in all of this, trying to simulate what's actually happening with the proteins and the COVID-19 virus. That sounds to me like it's a tremendous amount of data that's collected. Can you talk a little bit more about some of the challenges that you guys ran into when you were collecting all this data and trying to make sense of it and the ability really for you to crunch that data fast enough? Obviously, everybody was anxious to end the pandemic, so the quicker we get information, the better.
No, so it was a very interesting time, both scientifically and personally and technically at the beginning of the pandemic, because of course we were all dealing with the same things that everyone around the globe was dealing with, personally and professionally. But then with Foldinghome, we were in an interesting position where we'd been operating for 20 years, and we had 30,000 active devices around the world who were helping us to run these big calculations that are just intractable by any other means. And so we were regularly running these massive calculations and generating, data sets that even in their, stripped down and compressed form were, on the order of tens to hundreds of gigabytes, right? And so we were happy to share this data with people, but often at that scale, it was hard for us to just post on our website. And so we would end up, literally mailing hard drives around because that was the most efficient way to deal with the scale of data. And so what happened at the beginning of the pandemic, when we first shifted our attention and launched our first simulations of proteins from the coronavirus unfolding at home, is that people were all hunkering down at home, scared of the future and feeling pretty helpless and like they couldn't do much but hide. And we were able to come and offer people a legitimate opportunity to take part in going on the offensive against this new threat and trying to figure out, how does this thing work and how do we help stop it? And so within the first couple of months of the pandemic, we went from having 30,000 active devices helping us run simulations to well over 1 million devices helping us run simulations. And so, if you think about this, it's a pretty insane rate of growth, right? Within 3 months, we're nearly at 100x growth in the project. And so, at the first 5-fold growth, it was no problem. Like we're set up to scale to arbitrarily large scales. But in principle, right? But in practice, our servers started grinding to a halt from all of the communications. Our disk drives were rapidly filling up with these, many different simulation sets, each of which were hundreds of gigabytes. And so, in addition to trying to come up to speed on the science and be aware of what's going on and where the most high value areas that we could contribute we were trying to keep things from, falling to pieces under the load. And so we were, trying to deploy on the cloud, and we were trying to shuffle data around like mad to keep the disks from filling up on our on-premise servers. Our first tranche of data, again, even in its most stripped down and compressed form, is 200 terabytes, and that's probably a factor of 5 smaller than the intermediate state we were in. So it was quite the crunch. And extremely grateful that we got linked up with you guys, and you were up for stepping in and lending a hand.
Yeah, I think you said you grew initially you had 30,000 users that were signed up, and then it grew to 100,000 users. So each one of those users really being able to bring a certain amount of processing power, which is fantastic because that means you can take all of that data and process it more quickly. But the trick really was getting the data to be accessible instead of having to copy it around via hard drives and things like that, right?
Yes. So this was a major challenge, right? So one way of thinking about what we're doing is that we're trying to use all of these simulations to build a map of all the different structures that a protein can adopt, right? And so each simulation is effectively wandering around some part of this space of structures and sending us back GPS coordinates at regular intervals, right? And so what we're doing is really analogous to trying to build a maps app and having thousands of people explore different parts of the United States, sending us back data as they commute to work and run to the grocery store and whatnot. And then on our side, we're piecing this back together into our this map, right? And so the issue is that it's a lot of data coming back in. And sticking with the car analogy, if we suddenly have 100 times more explorers in our fleet all sending back data, we need 100 times more parking spaces and storage units for everything. And so the petabyte and a half that you were able to put at our disposal is just a huge boon, when we're sitting around with a bunch of RAIDs at 100 terabytes each that previously seemed really big, but suddenly seemed quite small given the 6 terabytes per hour that was flowing into our system, even in the first couple of weeks when we had grown to 400,000 users at that point.
Yeah, wow.
And Greg, there's a lot of things. Obviously, for those of us who've seen a picture of this, in the media, right? It's the gray ball with the little red fuzzy tips. It looks really cute. But through our first meetings, you talked about. I remember the demo Gorgon, right? And
Yes.
the ability to map this, and there's some really interesting numbers that come out of it. Things like being able to see something to the effect of a millisecond, and even you mentioned point in time, but just being able to map a millisecond versus you being able to do a tenth of a second, kind of explain what your ability to do those things really impacts the result that we can find.
Yeah, so starting out with the technical side, what happens with these simulations is we have a three-dimensional representation of a protein and its surroundings, and what we're doing over and 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 over again is asking, given how each of the atoms in the system pushes and pulls on the others, where are they going to be some small time in the future, right? And in order to avoid unphysical behavior like atoms jumping through each other, we can't ask, where is each atom going to be a second in the future? We're asking, where is each atom going to be 10 to the negative 15th parts of a second in the future, right? And so if we want to build up to, a second's time scales, it's like, a billion, it's more than a billion, times that we have to do this. So it's a lot of number crunching, right? So the upshot of this is that these calculations are extremely expensive. And so if you want to simulate just a microsecond, so 10 to the negative 6, a millionth of a second, then, that can easily take hundreds of years on a desktop computer, right? And so, using clever computer science and physics approaches and bringing many computers to bear on these problems and making use of GPUs, now we're at a point where we've made a major advance in the field where most papers in our field will have about a microsecond of simulation. Most good papers, a microsecond of simulation for a protein. And so what we were able to do with the scale that we were on, we're, at peak, even very conservatively, we had 5 times the performance of the world's fastest supercomputer at the time, all working on this one set of proteins from this teeny tiny virus that has far fewer components than the human body. What we're able to do with that is now say, okay, well, we're going to get milliseconds, so 1,000 times more simulation for each of these systems that we're looking at. And so looking at most of the proteins from the virus on this timescale, in aggregate, our first, major tranche of data has 0.1 seconds of data. So that's 100,000 times more data than a typical good simulation paper in our field. And so as you can imagine, this just gave us insight that was completely inaccessible to anyone else, without the access to this amazing resource that our volunteers in aggregate provided for us.
Yeah, well, since you bring up papers, Greg, Foldinghome has written a few papers here recently. Can you just talk a little bit about that so that the audience can go and read just a tremendous amount of how much you guys have contributed to COVID-19?
No, it's been really great. So the project has always been extremely productive. We've published hundreds of papers in many different fields over the year. And one of the exciting things about having so many people working together on this very pertinent set of closely interrelated problems with the virus is that, man, we were able to move really fast. So normally it'll take a year or two just to come up to speed with a new problem and to get the lay of the land before even taking on the first real deep steps in. In this case, in about a year, we were able to go from, wow, there's this new virus that we've never heard of before, to our first, major paper in Nature Chemistry, which is one of the premier journals in our field. And so this has been really exciting because, in addition to describing the world's first exascale computer, so the first computer that can do a billion operations per second, which is just insane. We were also able to get a lot of insight into how the virus works. And so one of our main foci, as Brian mentioned, is what we've come to call the COVID-19 demogorgon. And this is, the technical name for this is the spike. And so when you see these images of the virus, you've got this ball with all these spikes coming out of it. Each of those spikes is a complex of 3 identical proteins arranged with a 3-fold symmetry. And what really fascinated us here is that we know this thing has to move quite a bit, right? Because this is the protein that is responsible for attaching to proteins on human cells and initiating infection. right? And so to perform that function, as you can see in the images, it's out on the surface of the virus. And so this makes it a huge liability for the virus, because it's one of the things that our immune systems have a great chance of recognizing when the virus arrives in our bodies, and triggering a strong immune response that clears the virus before it even has a chance to get into any of our cells and really take off growing, right? And so the virus has evolved a number of defense mechanisms, and the one that we were most inspired by, if you will, is that the structure of the spike that you see in these images, it's actually closed up on itself, right? Kind of like a turtle having its head in its shell, right? And so this is a great protective stance for the virus, because it protects the most sensitive bits that need to engage with proteins on human cells from being recognized. But of course, being buried, that means that they can't actually, get at their prey. They can't eat like the turtle, right? They can't bind to this human protein called ACE2. And so in order to do that, this thing has to open up. So with this threefold symmetry, we kind of imagine this being like the mouth of one of these Demogorgon monsters from the television series Stranger Things. And we really wanted to know what that looked like, right? Because mostly what anyone has managed to see experimentally is this very common closed up structure. And so using our computational power, we were really able to see a lot of really dramatic opening up of this spike, which was fascinating, because if all you know is that it often looks like this, it's this surface that you have available to target with therapeutics. But if you know that what it looks like once it's opening up, you suddenly have all these previously unknown sites to target that we call cryptic sites. And fascinatingly, after we started sharing about this, on social media and stuff, and showing people data so that they could start, taking those ideas as quickly as possible, even before publication, some people did start reporting that they had discovered antibodies that neutralize the virus and surprisingly bind in these deeply buried regions that no one thought an antibody could get to. And so this was a nice corroboration, just as an example of one of our predictions. And we've done, similar things with many other proteins from the virus.
And I mean, that's all so cool. I mean, like learning that it's, the name of NSP16 and wanting to read those things. The one that really, as I was reading all the things that came through, and one of them got a lot of attention, was around this idea of phase separating with RNA. Like this year I learned what RNA was. We've always sort of known what DNA was, but I learned, everybody else knew what RNA was besides me. I learned what RNA was, but the idea, what is that paper about, the one about phase separating with RNA? And can you sort of explain that to us?
So this is a really cool area, right? Where, it's been common to think about proteins kind of like Lego blocks, right? And they go off and they attach to other proteins and they do different things. So a cell is this bag, that's full of proteins, and some of them play structural roles. Some of them are going around different parts of the cell, catalyzing different chemical reactions. And one of the themes that's been emerging in the last few years is that things are even more complicated than that, right? And that some of these different types of molecules can act kind of like oil and water, right? Where instead of being all mixed up and going about, randomly, for lack of a better word, in different areas as this well-mixed system, like oil and water, they actually separate. And that this is actually really important, and that biology has likely used this strategy to enable, compartmentalization of different functions. And so this brings up the tantalizing idea that, okay, well, we have these, viruses that are trying to invade our cells and replicate themselves. And so they're making all kinds of viral pieces inside of the cell. And if you think about them as randomly distributed throughout the cell, it's really perplexing. Like, okay, well, how does that then, all these random pieces assemble into working viruses, right? It's kind of like saying, okay, I'm going to dump a bunch of car parts in my garage, and then I'm going to shake it up. And, so you'd think everything is random, but despite your belief that things might be random, it's suddenly going to spring together into half a dozen working cars, right? And like, well, that doesn't sound quite right. And so, there's this tantalizing possibility that this ability to phase separate and have, things grouped together spontaneously is an important step in that process.
That sounds cool. It sounds like we need more exaflops of computing to figure that one out further.
That's right.
We do have a super great timely question from the audience, which is, how do you use social media to share information and in certain ways gain trust? And I think this specific article that was published on nature.com is a great example of that, right?
Yeah, I mean, this has been a really cool thing, and part of why we were able to have so much impact is that Foldinghome has been around for a long time. There's a lot of people that have participated the whole time. We're pretty active on Twitter and Facebook and our blog, and so we have good visibility. And there's a lot of people that have been part of Foldinghome. Maybe they were exposed to it in a high school science class where everyone installed it on a lab computer or whatever, right? Or they got involved at some other point and then maybe drifted away. And so this was really cool, is that in response to the pandemic, we knew normally one might be tempted to be a little defensive with scientific work at the bleeding edge, right? Like you want to make sure you do it and then publish it so you can get credit. But especially, we've been trying to promote, open science. And let's just make science go as fast as possible, and we'll work the credit and all of that out at the end. And so we really ramped that up with the pandemic, and we're, sharing information via Twitter and stuff, throughout, in the hopes that people would be able to put it to good use, and, sharing all this data online. And so that's been really great. I think people really appreciated getting some insight into what we were learning about the virus, and what steps we were taking towards enabling the development of therapeutics, and just seeing that all the compute was being put to good use.
Yeah, so Greg, to add on to that, we talk about sharing information. We've talked about just the massive amount of data that you guys are collecting. Can you explain in a little bit of detail the role that FlashBlade is playing in all of that work and what you're using it for at Foldinghome?
Yeah. So the FlashBlade has played a number of roles, and I have to apologize to you. I probably won't even succeed in listing them all, right? But one of the first things that's right up front is, like I said, we just had this data that we didn't know how to handle, right? And so just the magnitude of what you guys gave us was literally a lifesaver, right? Because otherwise we were going to have to start deleting stuff, or moving it into places where it'd be hard to retrieve. So that was the first phase. But it's not just a big hard drive, as you guys know. It's got a lot of extra capabilities in terms of compressing things and enabling you to work on things in place at insane speeds relative to what we had in place before with traditional spinning disks. And so what it's enabling now is for us to take some of our work on the algorithms for Foldinghome to the next level, right? Because what would happen in the past is we have these servers that are basically big RAIDs that bring in data from our volunteers and then store it until we're ready to operate on it, right? Until we're ready to analyze it scientifically. So a typical workflow would be that someone on the scientific team would set up 1,000 simulations, right? They would add them to the queue on one of these work servers, and they would kind of check on it every couple of weeks for 3 to 12 months, right? And at some point we would say, okay, that's probably enough data for the scientific questions we're trying to ask. So now let's copy that data over from the server to our computer cluster, where we have a bunch of nodes with lots of CPU cores and GPUs and enough memory to go and dig into this data and really analyze it. But in the meantime, what we've developed are these algorithms that we call adaptive sampling, where instead of just letting these simulations go for a long time and then getting all the data at the end, is that we're taking a more active role, where we start the simulations, we let them run for a while, and then we stop them and analyze all the data that we've generated so far. And this is this map building exercise that I keep referring to, where we're going to build a map of what we've seen so far, and then we're going to use that map to decide, well, where on this map would I benefit most from getting more data, either to improve parts of the map that I think are important, or to expand the boundaries of what the map covers, for example. And now let's go, start the simulations from there and get our next batch of data and keep iterating this. And we've been doing this for a couple of years now in our computer cluster, but for a variety of technical reasons, this is really hard to scale up for Foldinghome, because we need a lot more compute power to analyze all of this data that would come in from our volunteers on the fly. And so the FlashBlade has really been enabling for that, and so we've been re-architecting a bunch of pieces of Foldinghome so that we can have our work servers immediately bringing data into the FlashBlade, which is also connected to our heavy compute machines, so that we can analyze it there in place, make those decisions, and tell the work server which of the structures we found so far from the last batch of simulations or batches of simulations to start that next batch of simulations from, so that we can make the most effective use of finite computing resources.
And you actually mentioned part of this in your conversation. We're going to get back to it really quick, Greg, but we got another great question from the audience around people who want to get involved with not only just the business of this type of science, but also just like, what is it to get involved with AI? And so, Stacie, I'd love for you to be able to share with people, what do you think are the skills of the future as it relates to AI?
Yeah, so I would say it's a combination of, quite simply, getting enough data, right? Having access to enough data, and then just experimenting using different programming language and different applications, most of which are open source, that you can begin to stitch together applications and programming languages to bring in raw data, manipulate it in the way that you think might be beneficial to what your end result is going to be, and then come out and visualize that data. So I would say, in terms of skill sets, there's a lot of different courses out there online, and quite frankly, there's a lot of different models that are already available in the industry that people can experiment with. So I would say, roll up your sleeves, get out there, start experimenting with data. I'm going to make a plug for Kaggle.com, and I'm going to let Greg talk specifically about, because they do make their data. Greg, Foldinghome makes their data accessible to anybody who wants to experiment with it, and he can talk about where you can find that. But there's also a lot of great data sets out on Kaggle.com and different projects that are available. I think it's just as simple as getting out there, getting access to data, experimenting with the data in multiple different ways, and getting really comfortable with models. Like I said, a lot of models have already been developed, so it's not like you have to build it from scratch. It's just experimenting with those models and getting a sense of what they're capable of and what they're not capable of, and then pairing it with your data. And are you getting the outcomes that you would expect to see? That from a more industry broad perspective, that's what I would say. The skill sets are more around programming languages to manipulate the data and combining different data sets together is my personal opinion. And then I would say, last but not least, when we talk about the massive amount of data, Greg, you talked about it, getting data to the right place at the right time. I definitely think there's a place where the cloud, a big component of Foldinghome is actually even hybrid, where some data is going to land in the cloud and then some data is going to come back on-prem. And so making smart architectural choices that give you access to your data or portability of data is going to become more and more critical to organizations, and not being locked into one or the other is going to be really, really, really critical. So we see a lot of demand for what's really enabling that portability being around Kafka and Kubernetes, containerization, for example.
Yeah, we love these questions from the audience, so keep them coming. From my perspective, I love the ability to be able to architect, extremely large systems for performing enterprise AI and the actual biotechnology, science AI, and all the things in between. Like, AI has so many different use cases, and AI, machine learning, whatever we want to call it today. What I think is really cool about Greg's job, and the flip side of this is, and we get to talk to him a lot, these doctors who have these backgrounds in very unique sciences, who also bring up things that are heavily mathematical. Greg mentions adaptive sampling algorithms. that's not necessarily his it wasn't part of his thesis or dissertation, right? But it's really, maybe it was, but it's really cool stuff to see him combine those two techniques to get these types of outcomes. So Greg, do you want to talk about your perspective here?
Yeah, I think it's a really exciting time, and particularly in our field, kind of at the interface between machine learning and particularly deep learning and physics, because we have a lot of data coming in, and great desire to make sense of what's going on. So just as one example, we've been getting into deep learning to take all of these different data sets. So for example, we've got simulations of the spike complex I talked about from the SARS-CoV-2 virus, but also other coronaviruses like MERS and NL63 and the original SARS-1 virus, right? And so then we want to ask questions like, well, what's different about SARS-2 that makes it such a impactful virus compared to these other viruses that, while scary in their own right, did not become as much of a global concern by any means as the SARS-CoV-2 virus. And so what we've devised in another recent Nature Communications paper is a architecture called DiffNets, where we can take in structures from these different simulations, and through deep learning, we can basically coarse grain that data, like try to come up with a simpler representation of that stuff. And long story short, in the process, we're able to force it to say, okay, well, if you're going to do that network, you need to figure out what the most important differences between these different data sets are. And that's been an extremely powerful way of learning, like, okay, well, what is it that's unique about SARS-CoV-2 that distinguishes it from these other coronaviruses and helped enable it to become this global pandemic that none of us are going to forget. And I'm already preparing for my grandkids or whatever to ask me about what was life like during the 2020 pandemic for their essays in the future, right? And so this has been just hugely enabling, and it's so easy to get into with things like PyTorch, which are all publicly available, and data sets like ours that you can find on a service called OSF and through the AWS Public Datasets program.
Yeah. And Greg, by the way, when I tell my grandchildren what I was doing, I'm going to tell them I was hanging out with you, right?
All right, sounds good.
Folding at home.
We'll be referencing each other. This guy Brian helped save me.
So we really appreciate your partnership. We know that you're working with tons of great companies out there that are providing GPUs and CPUs and networking, and we're privileged to help power you with really fast, simple, unstructured storage for that same type of use case. It's been fun to work with you. And go ahead, Greg. Sorry.
No, I was just saying thank you. Same.
Yeah. And we want to challenge everybody. I still run folding at home on my computer over here. I literally can pop a breaker almost once a week. I'm looking forward to the winter, like when I could use it to help heat the home. They still have hundreds of thousands of users. They're not just solving COVID-19. They're solving all these other things. They mentioned Alzheimer's. Greg, I was thinking about you this morning. I read an article and it said something about heavy metals build up in the brain. And I'm like, does that have to do with a protein? Is it the protein's fault? So I was like, it's so exciting to have learned all these things about science that I did not pay attention to years ago. So I thank you for helping me, get past my poor education. And so again, everybody jump in. We're building science careers. We're solving problems. Greg is doing amazing things. The world's largest distributed supercomputer. You're going to see it again. They're going to set a record again, right? So really cool stuff. Everybody, thank you so much for joining us. Yep. Greg and Stacie, can't thank you enough.
So fun. Thank you, Greg.
Thank you. And the more people we can get involved, the merrier. Even with all the computers we have, we're still compute limited. The problems are that hard, and the results are that important. So we look forward to continuing to work with you and everyone else who wants to get involved.
Yeah, thank you.
Did you install it?